Welcome back, everyone, to The Building Blocks. I'm your host, Awade, and today we're tackling something fundamental: amino acids, peptides, and proteins. Sharon, I am so excited to dive into this. It's truly where life begins, right?
Absolutely, Wade. It's the bedrock of biochemistry. If you think about it, proteins are the workhorses of our cells, performing countless functions, and they all start with these small yet incredibly diverse amino acid units.
It's fascinating to me how something so tiny can build something so complex. I mean, we're talking about the building blocks, like literally. So, generally speaking, what exactly is an amino acid?
Great question. At its most basic, an amino acid has a central carbon atom, which we call the alpha carbon. Attached to this carbon are four different groups: an amino group, a carboxyl group, a hydrogen atom, and what we call a variable R group or side chain. And that R group, that's where all the magic happens in terms of diversity and function.
Ah, the R group. So that's what gives each amino acid its unique personality, if you will. The flavor, if you were making a protein stew.
Precisely. It's the distinguishing feature. And based on the properties of these R groups, we classify the 20 common amino acids into several categories: nonpolar aliphatic, aromatic, polar uncharged, positively charged, and negatively charged. Each category dictates how that amino acid will behave in a protein, how it interacts with water and with other amino acids.
That's so neat. So, like, some are water-loving and some are water-fearing. Is that where the hydrophobic effect comes in?
Yep, you're spot on. The hydrophobic effect is a critical driving force in protein folding. Nonpolar amino acids with their hydrophobic R groups tend to cluster together in the interior of a protein, minimizing their exposure to water. It's almost like they're trying to hide from the water, forming these compact structures, similar to how oil droplets coalesce in water.
I love that analogy, like a huddle. So, these amino acids, when they're just floating around, are they always charged?
Not always, but at physiological pH, which is around 7.4, most amino acids exist as what we call zwitterons. This means they have a positive charge on their amino group and a negative charge on their carboxyl group, making them overall electrically neutral but still dipolar. They're pretty versatile, acting as both an acid and a base depending on the environment.
That's so cool, like a molecular chameleon. And I know we have 20 common ones, but when did we even discover all of them? Because it feels like such foundational knowledge.
It's been a long journey. The first amino acid, asparagine, was discovered way back in 1806, but the very last one wasn't identified until 1938. So understanding these building blocks has been a gradual process of scientific discovery over many decades.
Wow, that's a huge span of time. It makes you appreciate how much goes into seemingly basic scientific understanding. So, once we have these individual amino acids, how do they actually clump together to form peptides and then proteins?
That's where the peptide bond comes in. Amino acids link together through a dehydration-condensation reaction, where a molecule of water is removed. The carboxyl group of one amino acid forms a bond with the amino group of another, creating this very strong, rigid peptide bond. What you end up with is a chain, with one end having a free amino group called the N-terminal and the other a free carboxyl group, the C-terminal.
Oh, so it's directional, like a sentence. It has a beginning and an end. And what's the difference between a peptide and a protein in terms of size?
Essentially, it's about the number of amino acid residues in the chain. Oligopeptides are relatively short, typically 2 to 20 amino acids. Polypeptides are longer, usually in the range of 20 to 100 residues. And when you get a chain of more than 100 amino acids, or often a functional unit composed of one or more polypeptide chains, we traditionally call that a protein. Though, of course, there's always a bit of overlap and flexibility in those definitions.
So basically, tiny chains are oligopeptides, medium chains are polypeptides, and big functional chains are proteins. Simple enough in concept, but I imagine getting these things out and isolating them must be incredibly complex. How do scientists actually study individual amino acids or proteins once they've been formed in a cell?
That's where purification techniques become vital. We have several powerful tools in our arsenal. One common method is dialysis, which separates molecules based on size, using a semi-permeable membrane. Smaller molecules can pass through, while larger ones are retained.
Ah, so like a molecular sieve?
Exactly. Another incredibly versatile technique is chromatography. We have different types, like size exclusion chromatography, which, as the name suggests, separates based on size, and ion exchange chromatography, which separates based on charge. Molecules pass through a column, and their differing interactions with the column material allow them to be separated and collected at different times.
That sounds like a lot of precision work. And then there's electrophoresis, right? I've heard that term before.
Yes, electrophoresis is another cornerstone technique. It separates molecules based on their charge to mass ratio when an electric field is applied. Molecules migrate through a gel, and their speed and direction depend on their charge and size, allowing for their separation and visualization. It's particularly useful for proteins and nucleic acids.
So, if I'm understanding correctly, each of these methods leverages a different property of the amino acids or proteins, size, charge, or a combination, to sort them out. It's like having a whole toolbox for molecular detectives.
That's a perfect way to put it, Aode. These techniques are absolutely essential for researchers to isolate, purify, and then study the specific functions and structures of individual proteins. Without them, our understanding of cellular processes would be vastly limited.
It truly is amazing how far we've come in understanding these fundamental building blocks, from a single discovery in the 1800s to being able to manipulate and study these complex structures in such detail. What's one thing you hope our listeners take away from today's discussion?
I'd say the sheer elegance and versatility of amino acids. They're simple in their core structure, yet the variations in their R groups allow for an incredible array of functions when they come together to form proteins. Understanding their individual properties is key to grasping how entire biological systems operate.
Beautifully said, Charon. It's like 20 unique Lego bricks, but with those 20 bricks, you can build anything. Thank you so much for breaking down such a complex topic into understandable pieces for us today. It's been incredibly informative.
My pleasure, Oade. Always a joy to discuss the wonders of biochemistry.
And thank you all for joining us on The Building Blocks. We'll be back next time with more fascinating insights into the world around us.